DNA stands for deoxyribonucleic acid. It is a double-stranded molecule with two strands that are twisted around each other to form a double helix. It is made up of many nucleotides. A nucleotide consists of a phosphate group, a ribose or a deoxyribose sugar, and a nitrogenous base. In a DNA structure, the two strands are antiparallel, meaning that one strand has the sugars facing the 5' end and the other strand has the sugars facing the 3' end. In DNA, there are 4 different nitrogenous bases, which can be split into 2 different categories, pyrimidines and purines. Adenine and guanine are purines and are double ring structures, whereas thymine and cytosine, the pyrimidines, are single ring structures. In DNA, guanine and cytosine are paired, and thymine and adenine are paired, both through the use of hydrogen bonds. C and G have 3 bonds, and A and T have 2 bonds. The enzyme that initiates separation of DNA into 2 single strands is called a helicase. It unzips the DNA and breaks the hydrogen bonds holding the nitrogen bases together, starting at the origin of replication. Once the helicase has unpaired the nucleotides, both strands can be used as a template to create double-stranded DNA molecules identical to the original in simpler terms. After the DNA is split, single-stranded binding protein stabilizes the newly single-stranded regions. The DNA gyrase is then used to make sure the DNA outside the replication site does not supercoil. A primer is produced under the direction of the primase at the replication fork. A primer is composed of a short sequence of RNA. The primer allows joining of RNA nucleotides that match exposed DNA bases at point of replication. Replication of both strands occur at the same time. However, the 3' to 5' strand replicates through a process called continuous synthesis. It is the leading strand, meaning that new nucleotides are consistently moving towards the replication fork, onto the new strands. The 5' to 3' strand replicates through a process called discontinuous synthesis. It is the lagging strand and is completed in fragments called Okazaki fragments, which are placed at certain points in the strand. An enzyme called DNA polymerase III allows addition of nucleotides in a 5' to 3' direction to produce a growing DNA strand between the Okazaki fragments to fill in the gaps. The process is shown below. Once that is complete, DNA polymerase I, another enzyme, comes in and removes primer from the 5' end and replaces it with DNA nucleotides. DNA ligase then ensures bonding between fragments and nucleotides to complete the replication process. The newly bonded nucleotides are called the new strand, and the original strand is called the parent strand. DNA replication is a semi-conservative process because half of the strand consists of the parent strand, and the other half of the strand consists of the new strand. The parent strand is always conserved.
In eukaryotic cells, transcription takes place in the nucleus due to the fact that DNA does not leave the nucleus. Transcription is essentially the process in which we go from DNA to messenger RNA, or mRNA for short. Note that the sense strand is the strand that the mRNA resembles when transcription is finished. It has the same sequence of nucleotides as the mRNA. However, in the mRNA, instead of thymine, uracil is present, which is represented by pink. The sense strand serves as protection for the antisense strand. The antisense strand is the strand that is transcribed, used as a template for transcription, in order to create an mRNA molecule that resembles the sense strand.
Okay, I'm recording.
The RNA polymerase molecule moves through and unzips the DNA. RNA polymerase reads the antisense strand from 3' to 5', generating the mRNA from the 5' end, attaching RNA nucleotides
You can send voice
to the
recordings over Facebook, you just
end
can't
as it
download
goes.
them.
It generates the mRNA from free bases in the nucleus. These bases are free-floating and then collected and used by the RNA polymerase to extend the growing RNA chain. It is important to realize the phosphate group, sugar, and nitrogenous base together make up deoxynucleoside triphosphates, or DNTPs. It is important to note that it will not transcribe every single gene possible in the chromosome, as this is not necessary. This is how cells specialize and differentiate. Lastly, note that RNA polymerase will zip the DNA back up as it goes. A base sequence that causes RNA polymerase to stop unzipping and detach is known as a terminator sequence, which serves as a termination signal to transcription. Thus, when the RNA polymerase reaches the signal, it detaches from the DNA strand, and the new mRNA strand is released. As mentioned before, the RNA polymerase zips the DNA back up as it goes. However, the few bases that were not yet zipped up causes the DNA to reform. The DNA is unchanged from this process. Before the mRNA can be used to make proteins, it needs to be processed. This may involve moving or adding sections of RNA. After a few modifications during this RNA processing, the mRNA will leave the nucleus and move into the cytoplasm. Here it will find a ribosome, and the process of translation will then occur. The initiation phase. The tRNA containing the matching anticodon to the start codon binds to the P site of the ribosome because the P site holds the tRNA carrying the growing polypeptide chain. The small subunit binds to the 5' end of the mRNA and moves along in a 5' to 3' direction until it reaches the start codon. The large subunit then binds to the smaller one. The elongation phase consists of increasing the length of the protein by adding more amino acids. Firstly, the mRNA is read 3 bases at a time, 1 codon at a time. The next tRNA, with the matching anticodon to the next codon on the mRNA, binds to the A or acceptor site. The A site holds the newest tRNA carrying the next amino acid that is to be added to the growing chain. The amino acids on the 2 tRNA molecules form a peptide bond. The ribosome moves 1 codon along the mRNA, which moves the first tRNA to the E or exit site to be released from the ribosome. The elongation phase continues in this manner until the stop codon moves into the A site. A polypeptide chain of amino acids is now formed. This is known as the termination or end phase. Phosphorylation then occurs. This means that ATP in the cell gives a phosphate molecule, or PO4, to the protein, which is what the amino acid chain is, which means that the ATP becomes ADP. Phosphorylation is important due to the fact that many metabolic reactions require ATP.